Good afternoon. Welcome to today's Wednesday afternoon lecture. It is my great pleasure to introduce you our guest, Dr. Roose. Dr. Roose received his MD and PhD degree from the University of Helsinki, Finland, in 1965 and 1967, respectively. He then joined the Brigham Institute in 1979 and served as the president of the institute from 1989 to 2002. Actually, I just learned today, Erkki has worked in Building 10 about 35 years ago and published a major paper, in Nature, primarily based on his work done in this building. So, Dr. Roose has made many important scientific contributions. His main contributions are in the field of cell adhesion. He was one of the discoveries of fibrolectin. His laboratory subsequently discovered in the fibrolectin the IRGD sequence and isolated cellular IRGD receptors, now known as integrins. The IRGD discovery has led to the development of drugs for many diseases, including cancer. Dr. Roose currently studies specific marker molecules in the blood and the lymphatic vessels that can be used to direct drugs and nanoparticles to tumors and other sites of diseases. Dr. Roose has published over 460 papers. He is a member of the U.S. National Academy of Science, Institute of Medicine, and American Academy of Arts and Sciences. He has received numerous awards. Just name a few. He is the recipient of the 2005 Japan Prize, the Jubilee Awards from the British Biomedical Society, and he was elected twice as the Innovator Award of Department of Defense Breast Cancer Program first time in 2002 and recently in 2008. Today, his talk is about his work on the vascular zip code in the targeted delivery of multiple functional nanodevices. Dr. Roose, please.
Thanks very much. I heard the day before yesterday a speech by President Obama, and when he came to the podium, he actually ran up the steps, and I tried it a little bit, but I decided that I don't have the spring in my legs that he has, and decided to walk up here. I'm going to tell you about vascular zip codes. And why I talk about vascular zip codes is that there's a tremendous amount of heterogeneity in the vasculature, in the endothelial lining of the vasculature. The blood vessels in different tissues generally look the same, but they are not molecularly the same. And of course, diseases then alter the vasculature as well. So this gives us a lot of targeting possibilities, and that's what I want to tell you about. So the way I got into what I now call vascular zip codes is that I got interested in tissue-specific metastasis. And being an adhesion researcher, I was particularly interested in the possibility that there would be an adhesion compatibility between the tumor cells that circulate and the vasculature of the target tissue they favor. And that would bring in more tumor cells into the target and give more metastasis. And if that were true, then the blood vessels in different tissues would have to be different. That's what we set up to study. And it occurred to me about 15 years ago that rather than using tumor cells to probe for that possibility of adhesion compatibility, maybe we could use phage libraries in live mice. And that's what we set out to do. So if you inject a phage library, and we use libraries that express random peptides. One can also express proteins and antibodies on phage, but peptides are the easiest to handle. And one can make libraries that have billions of peptides expressed in the library, such that each phage particle expresses one peptide. And the theory is, and it turns out to be correct in practice, that if you have that many peptides and are able to select the ones that have the activity you're interested in, there are always peptides in the library that do what you want them to do. And what we wanted them to do initially was to target a certain tissue. We started practicing with the brain and the kidney and showed that their vasculature was different, and that paper was actually published in Nature. So what happens is that the phage that express a peptide that has an affinity to the target tissue but does not bind elsewhere will concentrate at the target tissue. And we can isolate it from there, amplify it, repeat the process until we have until the library has collapsed to only peptides that have affinity to that target. We can also, of course, do this with a pathological lesion, and we're particularly interested in tumors. So here are some of the things that have been found, particularly using this method, but other people have used other kinds of methods with similar conclusions. First, every tissue appears to put a signature on its vasculature that makes the vasculature of that tissue unique, different from the vasculature of any other tissue. Some 15, 20 tissues have been analyzed in this way, and they all have specific markers, some of which have been identified molecularly. Tumor blood vessels are different from normal blood vessels. That was well known, of course, already before we started. Mostly the reason is that they undergo angiogenesis, and angiogenic vessels express different proteins and other markers than normal vessels. We showed for the first time that tumor lymphatics can be different from normal lymphatics. And I'll show you one marker to that effect. We also targeted wound vasculature and found specific markers. The idea was that, yes, it's angiogenesis in the wounds as just like in tumors, but perhaps it had a slightly different flavor to it than the tumor angiogenesis. And indeed, we found two peptides targeting tumors, targeting wounds, that while they do harm to tumors also, we have never seen them in our tumor screens. So there may be quantitative differences in physiological angiogenesis versus angiogenesis in tumors. And it's likely that other diseases put a signature on the vasculature. That's true of certainly of inflammation and atherosclerosis, and may be true in just about any tissue. It'd be worth studying using these methods. And what these markers make possible is to guide, to deliver imaging agents, drugs, and so on to tumors, and that includes nanoparticle targeting, and that's going to be my particular topic towards the end of the lecture. So, as I already pointed out in the previous slide, tumors have two kinds of circulation, like all other tissues as well. They have blood vessels, and tumors depend on their blood vessels. A tumor cannot grow without the blood vessels growing with it. And that's, tumors are good at inducing angiogenesis, the formation of new blood vessels. Generally, people say that a tumor cannot be larger than 1 millimeter in diameter before it requires blood vessels. Tumors also have lymphatic vessels. They're usually poorly functional, but they are an important, and they're not necessary for tumor growth, but they are an important conduit of metastasis. And it's the imbalance between the blood vessels, which tend to be leaky, and the lymphatic vessels that don't work very well, that makes tumors accumulate interstitial fluid, and they have a high pressure internally, and that becomes important as we go on with this lecture. Well, I don't want you to absorb this information. All I'm doing with this slide, which shows maybe half of the tumor-targeting peptides we have, that there are many of them. For blood vessels, lymphatic vessels, ECM in tumors, and plasma clots. Tumor vessels, because they are leaky, they leak fibrinogen and other plasma proteins into the interstitial fluid, and it gets clotted there by tissue factor, or the clotting cascade activated by tissue factor. And that's why tumors contain a network of clotted plasma protein, which normal tissues don't. And it turns out to be a great target for getting things into tumors. Now I'll have more to say about that in just a little while. Now, let me show you a couple of examples of blood vessel targeting in tumors. Here's one early peptide isolated by Kimmo Porkko when he was in the lab, and the receptor for it was identified by Sven Christian. Now, this particular peptide, when we find a peptide using the phage display, we then get rid of the peptide and synthesize it chemically and put a tag on it, and usually fluorescein. If we now inject the fluorescein-coupled peptide to a mouse that has a tumor, typically a tumor-targeting peptide accumulates in the tumor. This peptide shows some interesting features. It targets the endothelial cells in tumor vessels. Here's a good endothelial cell. The red staining is an endothelial marker. And you actually see that the dye has gone into the nucleus of that endothelial cell. But the tumor cell nuclei are also positive. So the first point from this slide is that some tumor-targeting peptides, they target primarily the vasculature, but then also the tumor cells. And those are the best kind of tumor-targeting peptides. They have both targets. The vasculature is important because it is available. The blood is in direct contact with it. And then, of course, it helps to get to the tumor cells, and you get more accumulation of the peptide in the tumor that way. The tumor cells bind this peptide in vitro also, and as you see from this confocal, some of the peptide is in the nuclei of the tumor cells. Now, it turned out that, and the point here is that this is what you'd call a cell-penetrating peptide. Only this is a cell-type specific cell-penetrating peptide. It only accumulates in tumors, not in normal tissues. Otherwise, it is similar to, say, the TAT peptide, which is non-specific. The receptor for this peptide turns out to be aberrantly expressed nucleolin at the cell surface. This actually is a common feature of angiogenic endothelial cells and tumor cells, that they put proteins that should be intracellular onto the cell surface, and they become targets there. And knowing then the target, we could take anti-nucleolin and first block the accumulation of the peptide in tumor cells, and actually the antibody, which I'm not showing here, accumulates in the cells instead. If we inject the antibody into a tumor-bearing mouse, the antibody accumulates in tumor vessels, whereas controls don't. So the third point is that once you know the receptor, you can change to any binding probe that suits your purposes, antibody, aptamer, whatever is suitable. Here are a couple of other peptides that target tumors, one that we call CAR. Now, if you compare CAR with F3, which I just spoke about, they're both cell-penetrating. They end up in the nuclei of the tumor cells, in this case, CHO cells. However, there's a CHO variant that is incapable of making heparan sulfate and other glycosaminoglycans. And these cells do not pick up the CAR peptide, but they do pick up F3, which doesn't care whether the cells make heparan sulfate or not, because it binds to nucleolin at the cell surface. So these peptides are cell-penetrating, cell-type specific, and exquisitely specific in terms of which cells they enter into. Here's a new one that we haven't published yet, that we call IRGD. Now, when Kazuki Sukahara and Tambet Tesalu, whose work I'm going to talk a lot about today, came up to me and said, we have a great new RGD peptide. I said, I've been working on RGD for 25 years. I don't want to hear about any new RGD peptide. And they were somewhat dejected, but did not give up, luckily, because it does turn out that this RGD peptide is very special. First of all, it accumulates in tumors specifically, as you see here. This is a transgenic tumor in the pancreas, and you can see that it picks up a lot of the fluorescein-labeled peptide, whereas the normal tissues don't. If you then do fluorescence microscopy, you see that the tumor is full of the peptide. And amazingly, even the phage that carries this peptide enters deep into the tumor, away from the blood vessels. If you take an RGD peptide, the one that is what we could call conventional RGD peptide, it does also target the tumor vessels. Here's a better picture of that with the phage. But it stays pretty much near the blood vessels. Doesn't do anything like the IRGD phage. And we can also use another type of a nanoparticle, not just a phage, in this case my cells, and they do the same thing and show the same difference. So here's a special RGD peptide that binds to alpha-V integrins on the surface of the blood vessels, but then does more. It is a tumor-penetrating peptide. Now I'll have more to say that later. Now the lymphatic vessels. We found this peptide. The work was primarily done by Pirjo Laakkonen, who was in the lab in the early part of this decade. And this peptide turns out to be a very interesting peptide in that it targets, rather than the blood vessels, the lymphatic vessels in tumors. And it's also extremely efficient. This mouse, which carries a tumor, I guess this thing died or is about to. Yeah. Do we have more batteries? So this peptide was injected as a fluorescein conjugate into that mouse 20 hours earlier than this picture was taken. And all we needed to do was to take the mouse and put it on a blue light table. No fancy imaging, no corrections for anything, no pseudo colors. The tumor glows, as you see there. If you inject the control peptide to another mouse, there's nothing there.
Maybe you can try use this mouse to see if that.
Okay. Well, if they can fix that, I'd prefer it, if you don't mind. Also, if you take normal tissues from this mouse, there's nothing there that one can detect at the level of microscopy or imaging. So, Valentina Vogel worked out the receptor for this peptide, and it turns out to be, again, the same thing as with nucleolin. This is a mitochondrial protein that for some reason tumor cells and tumor lymphatics and some macrophages in tumors put onto the cell surface. But even though, thank you, there are some normal cells that express this protein P32. It's just so faint that it doesn't work. Thank you. There are some normal cells that express this protein, but they don't put it on the surface, and that's in these panels, which I won't explain to you in detail. This protein, P32, is not expressed by all tumors. It's fairly reliably expressed by breast cancers. We surveyed 20 human real breast cancers, and 19 of them were positive at some level. But there are some tumors, like we've never seen this one in prostate cancer, that do not express it. One really interesting thing about this peptide, LIP1, is that it accumulates in the hypoxic areas of tumors. The red here is a hypoxia marker, EF5. And you can see that there's at the cell-to-cell level, they don't co-localize that much, but the areas that are hypoxic always attract this peptide as well. And the blood vessel markers are outside these areas, as one would expect of a hypoxic area. Now, the hypoxic areas in tumors are really important because they contain the cells that the drugs either don't get to or that the cells are resistant to drugs. And they also they contain cells, these areas contain cells that are genetically unstable. So to deliver drugs with this peptide may be very effective and do things that cannot be done otherwise. And while we initially thought that this peptide targeted tumor cells and tumor lymphatics, Valentina found that it also targets some sort of an activated macrophage that only exists in tumors. And that's shown here using various markers for lymphatic myeloid cells. So we actually think now that the lymphatic vessels are positive because these apparently bone marrow-derived myeloid cells become part of the lymphatic vessels. Sometimes we see lymphatics where every cell is positive around what appears to be a lymphatic vessel. Sometimes it's only one or a couple of cells that apparently are in the process of integrating into the lining of the lymphatic vessel. So this peptide may be a good way of delivering drugs to the macrophages in tumors, which are thought to be important for tumor growth. So I already mentioned to you that the apparent cell surface expression of p32 is important for the tumor specificity. There is always also elevated expression of this protein in tumors. If you, for example, look at the normal breast, there's almost nothing there, but breast cancers, as I mentioned, are regularly positive. And then this peptide, LIP1, also turns out to be an internalizing and tissue-penetrating peptide, just like the ones I spoke to you earlier. I don't have time to tell you about Valentina Vogel's recent work, which shows that p32 is actually an important regulator of tumor metabolism. And what it does is it promotes, paradoxically, oxidative phosphorylation. As you may know, tumors are known to have, to favor glycolysis, the so-called Warburg effect. This protein, which is elevated in its expression in tumors, actually counteracts the Warburg effect. And we're not quite sure what to make out of it, except to conclude that the Warburg effect may not be as simple as were originally thought, because this protein is also necessary for tumor growth. All right, so that's an introduction of some of our peptides. So I'd now like to show you what can be done when these peptides are combined with nanodevices in what we now call nanomedicine. So the idea of constructing nanodevices is that a device can do more than a simple drug. You can target a drug, but that's about it. You can't do much more with it. Whereas you can work in multiple functions into a nanoparticle. And I'll show you some of those functions and what can be accomplished with that. First of all, there's the targeting. And with the help of a resident Greek in the group, I coined that name, synaphic targeting, for this type of targeting, because people talk about targeted drugs. They are molecularly targeted. They only affect a certain target receptor. Our targeting is docking-based. We don't care what the receptor is as long as we can deliver elevated concentrations of our drug into the tumor. That's the difference. So synaphic refers to that. I don't know if you can read it out there. Now, one real problem with nanoparticles in medicine is that the reticuloendothelial system is built to do away with any foreign particle that circulates in the blood. And this remains a problem. We can use surface chemistry to avoid it, or at least mitigate the problem. We can also block the RAS, but there is no elegant solution to the problem because we simply do not understand how that system works, what the receptors are, and so on. And so that's an important ongoing subject of study. I'm not going to talk to you about subcellular targeting. That can also be accomplished with nanoparticles. I will talk much more about tissue penetration. And then here's a unique activity that we have built into some of our nanoparticles, and that is that they, having found their target, amplify their own homing at the target, sort of like what platelets do. If platelets find a breach in the blood vessel wall, they get activated, and that activation brings in more platelets, which get activated and on and on. And that gives you a blood clot, which may be a good thing or a bad thing, depending on where it happens and when. We can amplify nanoparticle targeting in an analogous way that I'll show you in a moment. We can build in diagnostic signals and drug carrier functions into the particles. We're hoping to and working on systems where we could give a command to the particles from outside the body that now is a good time to fire, in other words, release the drug payload. See, when you first inject nanoparticles into the body, or anything else for that matter, there is no gradient. It's the injected material is just going to be in the blood and everywhere. And then gradually, if it is a tumor-targeted nanoparticle or drug, its concentration in the tumor will increase and the rest of it will be eliminated. And there's a point where you have an optimal difference between the tumor and other tissues, including the blood. And that's when you want to have the nanoparticles fire. And we have some systems fairly far along on that, but not really completed. So we use different kinds of nanoparticles to accomplish different things. We started initially together with Sangeeta Bhatia, who is now at the MIT, with quantum dots. These are extremely bright organic particles that they are usually made out of toxic materials, and they don't have much potential in vivo. We're currently mostly working with iron oxide nanoparticles together with Mike Saylor, who is at UCSD, and again Sangeeta Bhatia. And we actually don't make them spherical anymore. We make them as nanoworms, and you'll see that word in some of my slides. We also make self-assembling micelles together with Matt Tyrrell at UCSB, and vesosomes, which are liposomes within liposomes, together with Joe Zazinski, who is also at UCSB. So I'll mention some of these particles. Mostly what I'm going to show you is going to be iron oxide nanoparticles or nanoworms. So how does this tissue penetration work? Well, we got a clue to it when Tambet Teosalu and Kazuki Sugahara were selecting libraries for cell binding using a prostate cancer cell line. And these T7 libraries, they express the peptide at the C-terminus of the phage coat protein. And we noticed over and over again that the libraries collapsed from 1 billion different sequences to these sequences that always had an arginine or sometimes a lysine at the C-terminus. And they were usually in a sequence context RXXR or KXXR or RXXK. So we figured that this sequence motif had to be some kind of something special, and it probably was some sort of a master internalizing sequence. And we started studying it, and indeed it was not only that the phage bound to the cells, it was able to take nanoparticles, here the phage, here quantum dots, into the cell, and again here's a confocal picture where there are a lot of particles where the nucleus is presumably inside the nucleus. So these peptides are internalizing as we had hypothesized. we then asked the question, is the C-terminal positioning of the arginine important? And the answer is yes. Even if the C-terminus is just blocked with an amide group on the C-terminal carboxyl of the arginine, there is no internalization of, in this case, quantum dots. And we can do that with phage also. We can also put an extra amino acid there, as we did here, putting an alanine at the end of the peptide. And what happens is that the peptide doesn't bind or internalize, but if we treat it with protease, then it does internalize. So these sequences can be cryptic in a peptide or protein and activated by a proteolytic cleavage that makes that arginine C-terminal. And then they become tissue penetrating, just as the peptides I showed you earlier. We can also design a peptide where we take the prototypic peptide and I forgot to mention to you that because of the requirement for this arginine to be C-terminal, we've named this the C-end rule. Some of you know, most of you know probably, that there's an end rule also for peptides that refers to intracellular degradation. Sender, we abbreviate this that way and pronounce it sender. Now, we can make a sender peptide by taking a prototypic sender peptide and putting urokinase cleavage site in it. Urokinase cleaves after arginine residues, so it restores the structure of this peptide, and they become tissue or cell penetrating in this case, as you see there. So these peptides can also be designed to be cryptic sender peptides. So now all we need to do is to put a specific homing sequence onto this kind of a peptide so that it homes into tumors, gets cleaved there, and becomes a sender peptide and enters into tissue. That's the kind of peptides I have told you about in IRGD and LIP1. They contain a sender sequence and then a sequence that binds to a homing peptide. So this would be in IRGD, the RGD sequence, and the receptor would be an integrin. Now, this first binding concentrates the peptide in the tumor. It then gets proteolyzed here and binds to a different receptor, which causes internalization and tissue penetration. That's how our tumor penetrating peptides work. And you've already seen this. That's how this terrific penetration in the tumors happens with IRGD. So what can we do then with further with these peptides? We can now couple a near infrared label onto the peptide, as we did here with IRGD, and we made them into micelles by putting a lipid tail on the IRGD peptide. They self-assemble into micelles that now are labeled with near-infrared dye. When we inject this kind of a micelle into a mouse that has a transgenic pancreatic tumor, some of them go into the liver, but a lot of them go into the tumor, which is impressively imaged here in the live mouse. If we take a control peptide, the liver gets more of the particles but there is nothing in where the tumor is. So it is highly specific. So we can do optical imaging. We can also do MRI imaging, as shown here. Now, the MRI pictures, if you aren't used to looking at them, they're kind of counterintuitive in that one is actually looking at reduction of the signal. So anything that is white lacks signal. So here's the pre-injection tumor, in this case, a prostate cancer. When we then inject IRGD, oops, there's a D missing there, IRGD iron oxide nanoworms, we start seeing black in the tumors, and it spreads with time into the whole tumor. If we use one of these conventional RGD peptides, we get a little bit of black signal, but it labels only the blood vessels and stays there, just like it did in the pictures I showed you earlier. If we don't target the nanoparticles, then there's no signal. So we can do MRI imaging also. We're working on a system that we call a hit-and-run assay. And this is a collaboration with Norbert Reich and Martin Moskovich at UCSB, who work on surface-enhanced Raman spectroscopy, nanoparticles that give you that SIRS signal. And so what we do is we take a mixture of nanoparticles, one of which is tumor-homing and the other not. And we inject them together. They go to the liver to the same extent, because that's not specific, but only one of them accumulates in the tumor, which changes the ratio of the particles, and we can then read it. We collect the blood from the mouse now, but we hope to be able to just put a detector on top of a blood vessel and then read it through tissue. And of course, the great thing about SIRS is that it has a lot of multiplexing capacity. One can barcode lots of particles. So this could potentially be a mixture of 10 different things or 20 different things that would each tell you a different thing about what is going on in the patient. We have this assay sort of working. A more sophisticated version of it would be if we take the IRGD peptide or some other peptide that gets proteolyzed in the tumor, we'd have a label at each end of the peptide. And now the end of the peptide that gets the sender sequence is internalized into tumor cells, and the other part of it, the C-terminal part, is released. So again, we might see a shift in the ratio. So much about diagnosis. How about therapy? I'll show you a couple of things about that. We've used a drug, a very successful drug that is a nanoparticle made out of Taxol or paclitaxel and albumin. And this, as I understand it, completely changed the way Taxol is delivered to patients. Instead of having to emulsify it and have it drip in over hours, this is a simple injection. Now we thought that maybe we can, if we target Abraxane, make it even better. And what I'm showing you is that, yes, we can. Here I am an Obama fan. We can improve the activity of Abraxane. And first of all, in this case, LIP1 Abraxane accumulates much more in tumors than the untargeted Abraxane, about 4-fold difference. And we can take a dose of Abraxane that has no effect on the tumor and show that when it is targeted with LIP1, it is active in preventing tumor growth. Work done by Priya Karmali mostly. We can also take IRGD and do the same thing. Here's the difference that we get between untargeted Abraxane and IRGD Abraxane. We have two conventional RGD peptides here for comparison. They may have a little bit of an effect, although this wasn't significant, but not much. The IRGD peptide alone has no effect. So again, we have improved the efficacy of, in this case, a nanoparticle drug. Of course, as I showed you, the RGD peptide and some other peptides of ours target the blood vessels, whereas LIP1 targets the lymphatic vessels and the hypoxic areas. So we're just now doing two-pronged experiments to see if we can further improve the Abraxane situation by targeting it both ways. Now, what is then happening here? Well, we don't understand it. We have a receptor candidate for sender, but even with the receptor, what is it that is happening? All we can say is that at the operational level, it seems that what we generate is some sort of a wormhole through cells and through tissues. And it seems to be a little bit more than a hole. Our feeling is that it's an active transport through these holes into tissue. And obviously, this is a big focus of our research. We took the term from the space theory that postulates that space-time can warp so that two distant places come very close to one another and one can fly through that wormhole. Of course, there are more mundane wormholes as well. So finally then, I want to tell you about the self-amplification of nanoparticle targeting that I mentioned earlier. Our system is based on clotting. We found that this peptide, which is a pentapeptide, cysteine, arginine, glutamic acid, lysine, and alanine, binds to clotted plasma protein. And I mentioned to you that tumors contain it. So it homes into tumors. And what happens when it homes into tumors when coupled to iron oxide nanoparticles is that it causes more clotting, which creates more binding sites, which brings in more of these particles and more clotting, and it keeps going. Initially, and we've published this is work initially done by Dmitry Simberg, we were only able to clot about 20% of the tumor vessels, and we didn't get a significant inhibition of tumor growth. We got great imaging results with this system. Now, Lilak Agemi has since been working on this and has improved the system so that now we do get inhibition of tumor growth. Pretty impressive, actually, here in a prostate cancer system, which is orthotopic, so we can't show you growth curves. We have to wait until we open up the mice and look at the end result. So that's shown here. And the key to this was that we stabilized the peptide against proteolysis. That improved its efficacy tremendously. That's why we get this result. Here's how much necrosis we get in the tumor, in the center, compared to a control tumor at the same size, no necrosis. Now the vexing thing here is that this tumor rim is always resistant. We've shown with ultrasound that we can actually block the circulation in this part of the tumor as well. So it apparently gets its nutrients from the normal tissue that is right outside the tumor rim. So that outer, the normal rim, as we call it in the lab, has become a big focus of ours. Hopefully those blood vessels under the influence of the tumor are also altered, and we can target them. But even without any drug, it works pretty impressively. And we can, of course, add a drug to this particle, and are about to do that. So what I have shown you is that we can target nanoparticles, we can amplify their targeting in a self-amplification system. I didn't talk about subcellular targeting. I showed you that we can get diagnostic signals from the particles we send into tumors, where they can have an improved drug activity, and they penetrate deep into tissue. We're working on the release upon command, and we're working on the RES avoidance. And finally, then, I mostly talked to you about work by Lilak, Kazuki, and Tambet. Ramana is our peptide chemist who makes all the peptides and their conjugates, Valentina, Priya, and some of the former members of the lab and collaborators. And on the background is my ranch, which overlooks the Santa Ynez Valley. Thanks very much.
With your internalizing peptides, like the IRGD, is there a limit to the size of the payload that you can add to those and still get it internalized?
There may be, but we haven't hit it yet. When we do this thing, we never see leakage of red blood cells. So at least it doesn't get through, it doesn't let through cells. The Abraxane particles are 130 or so nanometers in diameter. They seem to penetrate well, so I don't know exactly where the limit is.
Thank you.
Somewhere between those two.
Yeah, two small questions, one from each of your parts. In the first part is really an extension of the idea which you presented, that every tissue has not quite specific but distinctive zip codes. Right. Now, in one of your cartoon slides, I got the impression that the tumor may have a zip code and the same metastasized tumor may have a different zip code. Is that correct?
Well, it's more of a hypothesis at this time. We have one peptide that shows some selectivity like that towards metastasis specifically, or at least selectively, but we're certainly working on that. And a further question, similar question, is whether a metastasis from the same tumor in one tissue might have a different zip code as the same tumor in another tissue. And we're doing work on that as well. The important thing, though, at this point, from the practical point of view, is that the peptides like IRGD and LIP1, we know that they don't make a distinction between the primary tumor and metastases, and that's a good thing for therapy and imaging, of course. For theoretical purposes, what you're proposing and what I'm proposing would be nice.
Yeah. The second question concerns the nanomedicines or nanoparticles, whatever you want to call them. You set out to have two objectives of the nanoparticles or nanomedicine or nanodevices. One was the targeting, which of course you talked throughout the presentation. But there was also this objective of amplification.
Yes.
Now, to biologists, amplification tends to mean that there is some kind of self-reproduction. And obviously, these machines, little machines, are not self-reproducing. So it's not clear to me what was meant by amplification. Now, you gave the case of clotting. Now, clotting will increase the binding, but that's not necessarily the objective of amplification. To my mind, the objective of amplification is to spread the nanoparticles over all the tumor rather than just concentrate it.
Well, I guess we'll just have to differ about the nomenclature here, because I think if the particle creates more binding sites that brings in more particles, it is amplification of sorts.
But particles don't penetrate this. Let's take the case of solid tumors. Okay, even though the particles might be binding the vasculature, there has to be some mechanism for the particle to penetrate, to deliver whatever the payload might be, whether a gene or a drug or whatever.
Our penetrating peptides don't do what this Craker peptide does, but we may be able to come up with a system that does both.
So you don't imagine the clotting will restrict the movement rather than the clotting will restrict the movement of the particles going inside the tumor.
Right, into the blood vessels of the tumor, yes.
Thank you.
Yeah, I have a small question. It was an excellent talk. And
can you take the microphone a little bit lower?
Can you deliver the larger peptides, like approximately 20 amino acids in length, with the same efficacy, like what you did with the small peptides?
Right. I don't think there's any size limitation.
Okay.
We and others have made, for example, fusion proteins of, say, an antibody and a peptide, and they behave the same way as the conjugates and nanoparticles that I showed you. Why we like the short peptides is that the phage likes them.
Okay.
If you you can express a protein that has 150 or so amino acids on the phage and get away with it, but you have to work a lot harder to keep that kind of a library functional. But if the peptide is less than 10 amino acids long, and ours typically have 9 amino acids, 2 cysteines, and then a 7 amino acid stretch in between that is derived from the random sequence. They don't affect phage replication that much. So that's one advantage. The other one is that ultimately, just like happened with RGD, I expect these peptides, if they're going to be useful, to be converted to a different chemistry. And the shorter your peptide, the easier it is to do.
How about in the other tissues, like, for example, brain, or is it the same efficiency the peptides are delivered?
Well, we haven't done much work with the brain. In fact, the 1996 Nature paper we published is the only brain work we've done. And those were, at that time, we were satisfied with much less efficient peptides than we are now. So I can't tell how well it might work. But you're bringing up a potentially really important target. Because my prediction is that if you take this method and start targeting the brain, not only are you going to end up with peptides that target all of the brain vasculature and not other tissues, you're going to end up with peptides that go through the blood-brain barrier. You're going to end up with peptides potentially that target a certain region in the brain, certain nucleus, its vasculature, and so on. And then I fully expect that in diseases such as Alzheimer's, that the vasculature is altered. And amazingly, no one's really picked this up and done it. I'm into cancer, so I'm not going to do it. Yes?
I had a question particularly about your nanoparticles. So you were saying that you switched to elongated nanoparticles.
Yes.
First, how do you switch to selecting elongation over spherical?
Okay.
And thermodynamically speaking, I'm
Right.
guessing.
This was done in Mike Saylor's lab. And the simple trick turned out to be that if you take the iron oxide nanoparticles are typically coated with dextran. If you take short, low molecular weight dextran, out come spherical particles. If you take long chain dextran, they become elongated. And we found some advantages working with them to the what we call nanoworms, in that they seem to be probably because if you have a completely spherical particle and you have homing peptides on it, it's going to have a small surface interacting with the cell membrane. Whereas if it is elongated, the surface contact is longer, bigger. That's how we explain the observation that the nanoworms seem to bind better to cells than nanospheres.
And aren't you also getting better flow dynamics, I'm guessing, as a result, like in terms of your...
Yeah, I don't know. We have no data to support that possibility.
Thank you.
Okay, thank you very much for coming. We have a reception inside this building. Also welcome you go there. Okay, thank you very much.